and welcome to our EEG lab. I am Professor Louisa Kulke and I'm the head of Neurocognitive Developmental Psychology at Friedrich Alexander University Erlangen-Nuremberg. In our research, we use neuroscientific methods such as EEG, electroencephalography. And with this video, we want to show you how an EEG test session actually works. But what is EEG? EEG is a method to measure the electric currents that the brain produces from the surface of the head. By putting electrodes on the head, we can measure changes in voltage that are related to the brain activity from the top layers of the cortex. At the moment, we are inside the control room. This room is relevant for us to prepare the EEG system and to control whether we receive a good signal from our EEG system. As you might have already noticed, we have different hardware components in this room. The first thing that might pop to mind is that we have not just one computer, but several computers in this room. Why is this the case? The first computer here is our stimulus computer. This computer is there to present different images or auditory stimuli to our participant. And this is because when we measure EEG, we are usually interested in how the brain reacts to some stimulation. That's why we need this computer. The second computer over here is our eye tracking computer. In some of our studies, we use eye tracking to monitor where a person is looking on the screen and how big the pupil is. This can tell us a lot about what a person is paying attention to, what a person finds interesting, and also how aroused the person is at some point. Our eye tracker can measure eye movements and gaze positions with about 1,000 times per second. This means that it collects a lot of data, and this is why we keep the data on a separate computer so it doesn't interfere with the stimulus presentation. The third computer, you might already have guessed, is just there for EEG recording. With EEG, we can record up to 1,000 times per second from different electrodes. So if we record for a few minutes or even hours, we can have millions of data points on this computer. The problem with computers is the more programs you have open or the more they need to process, the slower they get. And you can imagine if we have these large amounts of data, the computers might get slow. You might even notice that at home, if you're working with an old PC and you have several programs open, it might get slower. And that might be annoying. Now, the EEG system works within milliseconds. And milliseconds are faster than anything we as humans might even notice. That means the PC needs to be so fast that we can't even notice how fast it is, which is why we need good hardware in this computer, and which is why we only run EEG software on this computer, so no other programs can interfere with it. Before the participant arrives, we can already prepare a few things in our EEG lab. The first thing is we can turn on our computers. We can then turn on the software we need for stimulus presentation. In our lab, we use Python or CyclePy to present stimuli with a high accuracy. We can start the eye tracking software on the eye tracking computer, and on the EEG computer, we start the software that we need for EEG recording. Once the computers are set, we also put other things here that we might need during testing later. We put some towels here for testing. We prepare the electrode gel that we will need during testing. We have a selection of different sized EEG caps here for testing. And when we test human participants, we try to very carefully abide by ethical rules so the participants get information beforehand and also need to sign an informed consent, which we already put over here. You can also already see our electrodes hanging over here. Electrodes are the sensors we use to measure potential changes from the head surface. And these electrodes are so-called active electrodes. When we measure a signal from the brain signal is very small compared to all the other signals in the environment. The active electrodes contain a little computer chip that allows the electrodes to amplify the signal as soon as we measure it from the head which makes it easier to measure the signal. All these things are already prepared and waiting for the participant. If we test infant participant, there are a few more things we prepare in advance. For example, we would warm up the gel so it feels nice and comfortable on the baby's head. We would usually have several experimenters present in the room so that the preparation of the EEG system works as fast as possible. And we will also have a selection of toys here to make sure that the kids never get bored and that the whole experience is as nice and comfortable as possible for the babies. So everything is prepared now, and we're ready and just waiting for the baby to arrive.